Welcome to the fifth installment of our Virology and Vaccine Institute of the Philippines, or VIP program webinar series. I am Madeline Ebarvia from the Environment and Biotechnology Division of DOST ITDI, and I will be your host for today. This two-day webinar is part of the Virology and Vaccine Institute of the Philippines, or the VIP program's information and education campaign on virology and other related topics. So amin pong isinusulong ang virology science sa Pilipinas. This learning session is made possible by the collaborative efforts of the following institutions: DOST Central Office, DOST ITDI, DOST PCHRD, DOST Picard, and DOST Balik Scientist Program. I am sure that everyone here is excited to learn. Am I right? Just a few reminder for our Zoom participants, kindly rename your Zoom account into this format: affiliation underscore full name. Again, affiliation underscore full name. And don't also forget to mute your microphones to avoid interruptions during the presentation proper. Now for this two-day webinar, we shall be learning a lot about bioethics on the use of animals in research, proper sampling, transport, and handling of avian species, which will be discussed by our resource speaker, Dr. Christina Laura M. Layson, a Balik scientist from the United States. Before we start, let's check first the participants, the number of participants here in Zoom. We are 151, and we have experiencing some problems on our Facebook stream, but we are now live in YouTube. So we will just be sharing our YouTube stream for this webinar, and we will try to fix some technical difficulties later on. So before we start with our new topic, let us have a short recap of our last webinars. So our first webinar was held last July 8 and 9, 2021, where our Balik scientist, Dr. Myra Josmelio, discussed about the bioethics on the use of animals in research, proper animal handling. It is followed by a back-to-back series of lectures from Dr. Cesar and Lourdes Nadala with the following topics: guidelines for research involving recombinant and synthetic nucleic acid molecules last July 15 and 16. Proper handling of multi-drug resistant bacteria last July 22 and 23. And biosafety and biosecurity in the realm of dual research of concern. So before we formally start today's lecture, let us formally welcome our distinguished speaker and their participants. So may I call Dr. Annabel V. Briones, Director of the Industrial Technology Development Institute, or ITDI. So Dr. Briones, the virtual floor is yours.
Thank you, Ms. Madel. Our distinguished speaker, Balik scientist, Dr. Christina Laura M. Layson, my ITDI family, DOST officials, colleagues, participants, guests, my warmest greetings to all of you. Today is the fifth webinar series of the project on establishment of Virology and Vaccine Institute of the Philippines, or VIP as we commonly call. I would like to welcome all of you here today and thank each of you for participating in this activity. This webinar series will run through next year, and we are fortunate to have 7 Balik scientists who will share their expertise in virology. The DOST VIP program was conceptualized to address the pandemic readiness of the country. The ongoing COVID-19 pandemic presents many concerns, particularly effective clinical and public health management, primarily on novel viruses. These issues can only be addressed using science and technology, specifically through research and development. Objectives of the VIP is 1, serve as the premier research and development institute in the field of virology, encompassing all areas in viruses and viral diseases in humans, plants, and animals. Second is act as a venue for scientists, both here and abroad, to work collaboratively to study viruses of agricultural, industrial, clinical, and environmental importance. And third is to establish strategic partnerships with the world's leading scientists, virology centers, and institutes, and conduct innovative and pioneering research that will advance the frontiers of virology in the country. To institutionalize the VIP, we get support from our lawmakers both in Congress and Senate by passing the bill regarding the establishment of VIP. While we are still waiting for the enactment of the VIP bill into law, the DOST has initiated several R&D projects implemented by ITDI for the Industrial Technology Development Institute, the St. Luke's Medical Center, and the Research Institute for Tropical Medicine, or RITM. These R&D projects are being implemented in partnerships with several local and international researchers and institutions. With these initial projects, we hope to build the capacity of the VIP and help resolve several of the pressing issues in the country brought about by viruses. Part of the project's activities is engaging the expertise of the Balik scientists to help us accomplish these R&D projects. Dr. Christina Laura M. Layson will discuss the bioethics on the use of animals in research, particularly in sampling, transport, and proper handling of avian species. I thank her for accepting the BSP engagement and helping ITDI in the VIP program. My special thanks to the VIP team of ITDI, the Technological Services Division, and the Planning Management Information System Division for organizing this event, the BSP Secretariat of PCHRD and PICARD, and the Balik Scientist Program, our ever-supportive Secretary Fortunato T. de la Peña, Undersecretary Rowena Cristina Guevara, Director Montoya of PCHRD, and Director Iborra of PICARD. I encourage the participants to join in the question and answer portion actively. I hope you will learn a lot from this webinar. Again, welcome and a pleasant morning to all of you. Thank you very much.
Thank you, Dr. Annabel Briones, for your warm welcoming remarks to our esteemed guests and our participants. So now to formally introduce our Balik scientist and resource speaker for this morning, Dr. Christina Laura M. Layson obtained her bachelor's and master's of science in molecular biology and biotechnology at the University of the Philippines, Los Baños, and Diliman. Thereafter, she pursued her doctorate in infectious diseases at the University of Georgia in Athens, Georgia. During her master's studies, she worked as a university research associate at the National Institute of Molecular Biology and Biotechnology at the University of the Philippines, Diliman. Dr. Layson was a guest lecturer and teaching assistant at the University of Georgia for the topics human anatomy and physiology, avian viral diseases, veterinary bacteriology, molecular virology, and introduction to virology. At present, Dr. Layson is a postdoctoral researcher at Exotic and Emerging Avian Viral Diseases Group at Southeast Poultry Research Laboratory, U.S. National Poultry Research Center, Agricultural Research Service, U.S. Department of Agriculture. Armed with her expertise in standard DNA, RNA, and protein laboratory techniques, including quantitative PCR, cloning, and protein expression, and knowledge on sequence analysis, phylogenetics, and homology modeling, she was able to publish more than 10 research papers in international peer-reviewed journals. She also served as a resource speaker in several conferences in the United States, Japan, and the Philippines. So now let's give a warm welcome and a big virtual round of applause to our guest speaker, Dr. Christina Laura M. Layson. Dr. Layson, the virtual floor is yours.
Hi, good morning. Good morning. Magandang umaga po at maayong buntag po sa inyong tanan. Welcome po sa talk for today. Thank you, Dr. Briones, for that introduction, and also Madel. Thank you so much. Okay, so let me just take a moment to share my screen. Okay. Can you see my screens okay?
Yes. Yes.
Yes. Okay, great. And I guess you can hear me. As in that. Great. Okay, so today I will be talking to you about the care and use of avian species with a specific focus on sampling, transport, and handling. Can you see my, I forgot to ask, can you see my slides or my presenter notes?
We are seeing your slides.
Just the slides. Okay, perfect.
Yes.
Yes. Okay. All right, great. So let's get started. So I wanted to start with sharing a little bit about myself. I guess Madel already shared to you like all the CV, the resume type, but I wanted to just take a moment to also mention why I got into virology, because I think a lot of you may be interested why I went into virology, how I went into virology, and it all started with this bacteriophage, and it's on the this diagram on the left. It's something that I learned at while I was an undergrad at MBB, and it was on molecular genetics, and it was the life cycle of the bacteriophage. And then it was just one of those lessons that I just stuck to my mind, and how amazing the viruses are, and how it's such an elegant biological agent, how this small particle can do so many things to the cell. And then as an undergrad, I had the opportunity to work at a virus lab. And then I never turned back, really. So as an undergrad, I worked with abaca, which is yung banana-looking plant, the upper right. And then for my dissertation, I went and worked with chickens. And right now, I still work with avian species, and that's why I'm giving this talk on avian species. So that's one thing about myself, but I also wanted to hear from you. So I'll stop sharing my slides and pull up another set of slides called Mentimeter. Hopefully this will work. I hope you will indulge me in this effort. So let me share another set of slides. Can you see the screen? Okay. Can you see the slides? I think you can, because I see it.
Yes.
Okay. Okay. So what you do is you go to www.mentimeter.com and put the code 33066117, because I'd love to hear from you. So let's just see how this goes. Hopefully it will work, because I wanted to make this lecture interactive. So question number 1, how are you feeling today? Ayan, there's people coming in. Question number 2, very important for Filipinos. Do you eat rice in every meal? Question number 3, outside research, have you worked or handled avian species? So this may include maybe growing up, you had pet chickens, maybe, or you have other pets or alaga in your household. And then 4th question, for research purposes, have you used or have you worked with or handled avian species? Okay. Numbers are coming in. That's good. we'll give a couple of minutes. I'm feeling excited today. I'll answer the questions while people are feeling excited today. So that because I'm sharing this lecture with you, hopefully we'll make it fun. Do I eat rice in every meal? I don't. You know, I eat once or twice a day. Yes. Brown rice is trying to stay healthy. And then outside, obviously, I work with avian species. I'm not allowed to have pet birds because of my job. It's something that we don't do. So I'll give it maybe 30 more seconds. I think it stopped moving. Going once, going twice. Good, great. Thanks, guys. Thanks for indulging me with this, with this effort. So, people are feeling pretty good today. 3.7 out of 5. I say that's pretty good. Do you eat rice in every meal? 3.58. I think almost, maybe 2 to 3. That's good. Pinoy talaga kayo. Okay, and then outside research. This is actually the questions I really wanted to ask. Outside research. People have handled, maybe some have handled avian species, but even fewer have handled avian species. So that's good. Sort of what I expected. Avian research on avian species is kind of a niche. It's very specific, sort of a niche, but very important. You'll see later why. Okay, next question. I had the next question. If I can get it to move. Oh, what kind of, maybe you can just put it in the chat. It's not advancing. What were the species that you work with? I'd love to know what kind of species you were working with. Yeah, maybe in the chat if you're willing. Good. Okay, we've got mallard duck, itik pinas. Great. Mallard duck. I also worked with mallard ducks. They're great to work with. Anyone else? And that's from Pauline Saludo from UPLD. Okay, I'll stop sharing. Going once, going twice. Maybe that's just one person. That's okay. Great. Great. Thank you so much for that. Yeah. Okay, so I'll go back to the real slides. Oh, I see. Okay, can you see my slides okay? Yes, no, maybe put in the chat.
Yes po.
Great. Okay. Great. Okay, thank you so much for that. I actually wanted to really just test also if it works. Thank you for that. Okay, so I'm meant to meet your questions. Okay, so for today, I would really like to have these two main learning goals. So one is to gain understanding of the principles that guide the use of avian species for research purposes. And then the next is to enumerate the steps that are necessary to successfully to have a successful research project involving avian species. And then for this, we're having, since this is a two-part lecture, we'll have the first part will be, I've named the planning phase, and the second part is the execution phase. So actually, you'll see later there's a lot of planning involved with using animals for research, and you'll see why. So there's two main things in the planning phase. One is animal care and use program, and the other is animal biosafety. So we'll be talking about that today, and then on Wednesday we'll be talking about more specific stuff on like how do you just getting to know what are avian species in general, just a brief overview, just the relevant stuff that's important when you do research with them, and then other very specific, more technical stuff. So today will be more on the planning, which is which can get very involved. Okay, so why do we work with avian species? So many avian species are important in agriculture. I think the majority of the work that's done on avian species are from oh, somebody did migratory shorebirds. Oh, that's great. Okay, sorry, I was looking at the chat. Okay, many avian species are important in agriculture. So you'll see in the manuals that a lot of the guidelines are for domestic poultry like chickens. In the US, we have a lot of turkeys, but I would assume in the Philippines, there's more quail, ETIC, like Pauline mentioned earlier, and someone actually said migratory shorebirds. That's great. Yeah, so a lot of the material that you'll see here are actually on chickens, just because there's so much more material on that, but it also applies to other avian species as well. So avian species are also experimental models for certain diseases. So for example, for our work at Southeast Poultry, we have the mallard model as a model for avian influenza in waterfowl, which is the reservoir for avian influenza, and that's why we work with waterfowl a lot. And then for avian species, the research work can be on nutrition. That's more on the management side. Some do lighting, or they do interventions to reduce, let's say, the salmonella loads, more on a public health, human health perspective. Of course, there's pathogens, which is my favorite because we're also at the VIP, the Institute of Virology seminar, and also for ecology and conservation. So maybe Ralph Pino from IDTI worked on migratory shorebirds. So yeah, it can also, working with avian species can also do have something to do with ecology and conservation, and also toxicology. So this is a unique one. There was a seminar that I've attended before that worked on pigeons, because pigeons are peri-domestic. So peri-domestic species, meaning they live in close proximity with humans. So you can some people use pigeons as proxies to look at toxic materials, or like lead or other environmental toxins and chemicals. I'm not a toxicologist, but anyway, toxins that are in the birds, just because they live so close to us, and they also live in a relatively small area compared to other birds. So that's an interesting trivia on pigeons. So the planning phase. So I really like this quote. It's from the 7 Habits of Highly Effective People. It's begin with the end in mind. So when you can think of it as an animal experiment or a research activity involving the actual animals. That's actually like maybe the easiest part of it all. I think the hardest part is figuring out what to do and planning and getting everything together. So you can think of it like as a show, like your favorite noontime show, or maybe also the Olympics. They prepare so much. They prepare 4 years for 4 minutes, let's say, of a gymnastic routine, or they prepare so much, a couple of days or maybe weeks, maybe months, just to have a movie that's 2 hours long. So the animal experiments are also kind of like that. So there's a lot of effort that's done beforehand before you actually do the experiment. So we'll talk about that today. Okay, so first off, the first part of this planning phase is animal care and use. So what is this animal care and use? And you folks might have also heard of that. So the animal care and use program are the policies, procedures, standards, organizational structure, staffing, facilities, and practices that are put into place by institutions to achieve humane care, that's a typo, and use of animals in the laboratory throughout the institution. And this is a direct quote from the Guide for the Care and Use of Laboratory Animals, which is a manual that's like quote unquote the Bible for laboratory animal research. So it has a lot of guidelines associated with that. Okay. And so one group of people that are involved in making sure that the animal care and use program is put together well is the IACUC, or the Institutional Animal Care and Use Committee. So they primarily do assessment and oversight of the animal care and use program. And they also play a key role in interpretation, implementation, oversight, and evaluation of the animal care and use program. And this is because there is really no, so like, remember, like the manual actually just said guidelines. So there is really no step by step, like very specific, like this is what you need to do. I mean, outside of the confines of law too. So animal welfare laws, included, but there is a lot of flexibility, or maybe not flexibility, but more there's the implementation of the animal care and use is really dependent on what the research needs are, what the resources available are. And so the job of this group of people is to decide on how they're going to do the animal care and use. And the members of this group include scientists, veterinarians, non-affiliated scientists. So this means that scientists that are outside the institution. So there's a by outsider expert view, and also community members. So there's also one person or one or two people in the IACUC that's a community member that's not necessarily a scientist, but we also, as scientists, we often use like taxpayers' money, right, for our research programs. So we do have a responsibility to have to seek feedback from the community on whether or not they think that our research is relevant, or the way that we do our animal studies are according to the values of the community. And so this IACUC committee is regularly meets to assess and evaluate existing animal care and use programs. So that's key, because it's not a one and done thing. It's something that is refined and always regularly revisited. Okay. So the planning phase and the animal care and use. So before starting a project, and this is from our lovely attending veterinarian at Southeast Poultry, and Hurley Bacon. So before you start a project, these are her guidelines, and which I think are really good. So first is become knowledgeable about and conduct all activities involving the care and use of animals in accordance with regulations, guidelines, and approved policies. So be just aware of what programs exist in your institute, and all of the regulations and guidelines, right? So first, be in the know. And then the second is to design experiments based on scientific merit. Again, compliance with animal care and use policies. And also meeting the goals for animal use, called the 3Rs. So we'll talk about that later. And the procedures that will minimize animal pain, distress, and discomfort. So this is like a funny meme from her. Your poor planning is not my emergency. So they really want you to think ahead. So very important. Begin with the end in mind, way in advance. Know what you're getting into and whether or not, so that we can all so we can all do our research with care and for the animals. Okay, so, and then this is a continued list. Prepare the actual care and use protocol. So this is an actual document that says, number one, I will go sample the birds at this time, or I will get birds from this source. They will be brought into the facility. They will be quarantined, etc., etc. So you will write in paper, in black and white, submit it to the animal care and use, the IACUC committee for their approval. And then once that's all approved, your responsibility is also to ensure that all personnel qualify for the duties that they're assigned to. So everybody is knowledgeable, is trained, and is competent in delivering those the care and also doing all of the research procedures. And then conducting thorough orientation for our personnel, so that includes animal care and the investigators, and also maintaining complete records of all the procedures that are undertaken. So the IACUC doesn't just do the approval of the protocols, they also do some oversight. So you do have, when you finish a study, you are required, or at least for us, we are required to submit our records. So that may include like the room notes. So notes that you did while you were doing the research, and and if there were any adverse events or unexpected events, that also needs to be reported. And this is for evaluation purposes. So remember, this is not, the IACUC is not one and done deal. So it's a continuous process that we always undertake. We always seek to improve. Wait, great. So we talked about earlier the 3 R's. So what are the 3 R's? That's replace, reduce, and refine. And these 3 principles guide whether, so you ask yourself when you think of a scientific question, what are the 3 things, these 3 R's, should I really use animals for this research? So the first one is replace. So ask yourself, like, is it absolutely necessary that I use animals to answer this question? So for example, if you're, let's say, trying to find a new vaccine for COVID-19, for SARS-2, and you've made your construct, let's say you've made your vaccine, and you wanted to test it in animals. And so in that case, I think there is really no substitute for animals, because it's the mammalian body is such a complicated system, right? Yeah, there's the immune system and different respiratory system, etc. But there are some instances where you can answer questions just using either computer simulation, cell culture experiments. So that's number 2, we can reduce the number of animals that we use. So for example, for our facility, we also take note of how much animals we use every year. So we always seek to use the absolute minimum. And this can consider what type of species that you use. So typically for a mammalian model system, so like there's a mouse here, I understand there's a mouse here, but this applies to avian research as well. The mice that are used for the lab are what's called inbred mice. So they're genetically very similar to each other. So, but so they can get away, because they're very genetically similar to each other, they can use smaller groups of animals because they're inbred. But for avian species, a lot of them are outbred. So unless you have a very specific cell lines, which are very few in the world, there are very few genetically the genetically similar lines in that are avian. But generally, for example, in avian research, we typically use larger numbers because the species are not outbred. So you also take into consideration for that. So you use more, but we use the minimum that can answer your question. And last but not least, refine. So we always seek to improve our care for animals, and there's always an update of training, and you read the guidelines every year because they're refined every year. And at least that's true for us. So we have to do our IACUC training every year. Okay, so what are the responsibilities of researchers? So one is to ensure the humane care and use of animals, and also have a direct continuing responsibilities that they're cared for and used well. And so I underlined in bold here just to emphasize that research using animals is a privilege. So it's not a right. It's not a right. So it's a privilege, and it comes with responsibilities and some accountability. So we are this is not an activity that everyone should do just because it's for fun. We don't do it for fun, or we don't do it because it might be interesting. We do it because it's necessary. It's necessary to answer questions. For example, the example would be finding a vaccine, or maybe screening for drugs that can be effective, let's say for a virus. So really important. It's a privilege, so we should take care of our responsibilities for that. Okay, so yeah, so the next thing is for training of the personnel. So all persons should be adequately trained and qualified for the activities that are undertaken for the care and use of these animals. And this can be gained through lectures like this one we have every year, and also hands-on activities, so which might not be very possible during a pandemic situation, but we also have participate in hands-on activities just to make sure that we have at least within the institute fairly standard procedures. So we all kind of do the same, and also share some tips. That's also nice to talk to other people who do similar work. And again, it should be practiced and refreshed regularly, ideally every year, if you can, if possible. Okay, and then one other thing I wanted to mention in the planning phase, in this part, is before starting your project, at least for us and for in practice, we always have a powwow. We always have, like, no matter how much experience the team has, we always have a meeting before you start an experiment, just to make sure that all the T's are crossed and all the I's are dotted. So animal research involves a lot of moving parts. There's the investigators, the veterinary and animal care people, and the supervisory personnel. So all of these people need to come together and make sure everyone's on the same page. And then the things you talk about are like, what's the schedule look like? Do we actually have the rooms for it? And even close to, and the more detailed it is, the better, because once you're in the actual doing the actual experiment, it's really hard to think. We like to think like your IQ is like 50 when you're like in the moment, but if you've already planned way ahead, you've already thought everything through, then it'd be so much easier, and it will lead to fewer mistakes, and fewer mistakes means that we can use less animals, less resources. You don't have to repeat any experiments, and you get the results that you need. So we're more productive. So really important, plan ahead. And then the duties, and we also like to talk about what are the responsibilities of each person. For example, who will go check on the animals, who will label the tubes, which is very important, very maybe a menial job, for some, but absolutely important. Labeling tubes with nice handwriting, if it's handwritten, if it's printed, much better. But labeling of tubes and the baggies for the samples is super important, because they all look the same. Really. And then necropsy duties as well, and preparation of materials. You can also talk about that during the meeting. Okay, so that's the animal care and use part. So the next part is focused more on biosafety, because safety first. So again, this goes back to research is a privilege, right? And it's not something that's done every day, by everyday people. It's done by qualified people in a facility that's appropriate for the activity that they are doing. So the number one thing is we want everyone safe, and that includes the people who are working, the lab people, the environment, the greater community, and also the sample. So we'll talk more about that as well. So So again, just so I know, Dr. Cesar also talked about this, but if you were there, this will be a review for you. But biosafety is a combination of factors, and also like to use an onion as an analogy. So it's multiple layers of protection. So there's layers upon layers of protection. So if there are any vulnerabilities, if we have more layers, then there's likely to be multiple breakdowns in layers of protection. So this can be, if that includes you as the person who are working and your coworkers, the facilities, the procedures, and the practices within the facilities, and the actual physical and primary and secondary barriers that are in use. So before you do anything else in terms of biosafety, risk assessment must first be conducted. So risk here is really the marriage, of probability and consequences. So that's on the diagram on your bottom right. So what is the likelihood that this is going to happen, and what is the severity of the consequence? And we want to keep all of that minimum. So low probability, low consequence, as much as possible. And then this takes into consideration routes of exposure. We'll talk about that in the next slide. The availability of interventions and vaccines that mitigate or prevent disease. So that includes knowing what pathogens you might be exposed to, what hazards you might be exposed to, and how we can mitigate that if something happens. And also the current constraints available, like, do we have the budget to do this? Do we have the facility to do this? Do we have the expertise to do it? These are all important things to consider when you do risk assessment. And so risk assessment is undertaken by management, the leadership, and the biosafety people. So again, this is also this is done by a group of people, not just like one or two people. And it's also continuously evaluated. Okay, so routes of exposure. So here's just, this is from Principles of Virology. And can anyone, I'd like to keep this, can anyone put into the chat what types of pathogens you can get exposed to by respiratory route? So either through the mouth or the nose. Let's see if I can see in the chat. Are you still there? Are you still alive? Respiratory routes. Pathogens that can be inhaled through the nose, or yeah, anyone there? You can put it into the chat. Airborne bacteria, viruses and bacteria, very good. That's from Brandon Allen, Bellamy, Brandon Allen Martinez, good. Like SARS-2, right? The causative agent of COVID. And then how about an example of pathogens that can be ingested? You can put it in the chat. Yeah, E. coli, great. E. coli, yeah, that's good. Salmonella, it's a good one because that's an important one that comes from avian species. Good, great, thanks guys. Staph, yeah. Staph aureus, yeah, great. Okay, so you guys are all experts on routes. Yeah, so routes of exposures, like considering routes of exposures are really important. So that we can use the proper PPE, which I think a lot of people are more aware of because of the pandemic. So here are just some of the PPE examples that you're probably familiar with. So this first one is the glove. So I like this picture because it's actually showing you how to take your gloves off properly. So this is like the beak method. If you don't know it, Google it. And the beak method is like a very safe way to take gloves off, because theoretically the outside of the gloves are dirty, right? And the inside is clean. So you want your skin to never be exposed to the dirty side. And this is true. We do our biosafety officer does make us practice taking gloves off in front of him, and he puts like I don't know what it is, but it's a cream on the outside to simulate like bacteria, and it glows in black light, so you will know if you got some on your skin. It's pretty safe though. It's not bacteria. It's not real bacteria. Okay, so other stuff that's in here. Does anybody know like this second? Can you see my mouse? I'm not really sure if you can see my mouse, but can you see like the second column bottom picture, the object that's there? Does anyone know what that is? So there's like the surgical mask, right? And then the one in the bottom. Does anybody know what it is? Yeah, N95. Yeah, the other one's a facial, the second column. Yeah, so the N95, does anybody know what N95 means? Any takers? Maybe bonus points. I'll give it a couple of seconds. Yeah, so the other two are like the safety glasses. So the real difference is, yeah. So the difference with the regular glasses, which I have on, and safety glasses is the sides and the top are closed. So you can see here with the top, the sides are closed. So if there's any splashing that comes from the side, then it's prevented from getting into contact in your eyes. And then same with like the face shield, it's more around your face, so you'll protect the face a lot more. Great. Yeah, so yeah, N95. Great, you guys are experts. So yeah, someone put in N95 means that filters 95% of airborne particles. That's exactly right. And then the last PPE is a PAPR, which is battery operated. It has a filter, and what it does is from the filter that's strapped on your waist, it blows filtered air. So the same kind of idea with the N95. It's not exactly an N95 material. It's called a HEPA filter. There's a HEPA filter there. It filters the air from the room and then blows it through the hose and on your like over your head, and because the hose is actually sort of at the back top of your head, blows air away from your face. Yeah, it's a NIOSH. NIOSH is, I forget what it was, but it's an accrediting agency. N95 is yeah. Okay, anyway, so a PAPR blows clean filtered air away from your face. So there's positive air pressure blowing away from your face. So it filters, yeah. So that's how it works. So that if there's any contaminated air, it gets blown away from your face. That's really important. It flows away from you. Okay, so that's for PPE. And then one other concepts that I also wanted to discuss is cleaning, disinfection, and sterilization. And these three activities are important when we do our experiments because we always do them. So we always at the very minimum clean and disinfect before and after after any procedures, right? So cleaning is, we all know cleaning, it's the removal of foreign materials, and it's usually done before you disinfect and sterilize, especially stuff like reusable stuff like scissors, the PAPR, the hood, the hood that was or that I showed you earlier, that's reusable, so that also needs to be disinfected and cleaned. And then cleaning is usually done with detergents and enzymatic products. And then for disinfection, this is the reduction or elimination of agents that might be harmful. And then usually we use chemicals for that.
For the starter project, CNN and they provided project overview last week. and I also focus on test environment and installation of the tool and requesting access. So last Thursday, TVS account granted to me, and I started check out the regression test for
I'm sorry, I actually can't hear you very well. Are you asking a question?
for the large file, for the timeout error in the large file. So the workaround I have done is to increase the connection timeout from by default it's 60 seconds. So I increase it to large number to avoid the timeout error in the large file. So checkout was done. And for this week, I will focus on the regression testing organization for the DBE tests.
Hi, is the person asking a question? I can't understand you. If you are asking a question, put it in the chat. I actually can't understand you very well. If you're not asking a question, please mute your phones. Mute your microphones. Thank you. Okay. Where was I? Yes, disinfection. Okay, so yeah, and one thing that someone actually asked me, disinfection, there is a minimum contact time. So, and it needs to be evaluated depending on what agents you're using. There is a minimum contact time. So, for example, bleach, for example, we use like a 10-minute contact time. It's not that you, let's say, that's why when, you know, the messaging for for washing your hands is 20 seconds minimum, right? So 2 happy birthdays, because there is a minimum contact time for disinfection. So always follow the minimum contact time when you use disinfectant. And then last but not least, sterilization is destroying all forms of microbial life. So this is usually done by physical and chemical methods. So there's an autoclave on your bottom right. And for that. And I also noted here that the methods for disinfection and sterilization before should be verified before you implement it. So make sure that it actually works. So I guess this comes back to planning. So for example, I'll give an example. So for example, influenza is an envelope virus. And that means that it has a lipid outer membrane. So, and because it has lipids, it can be easily inactivated with detergents or alcohol. But for example, picornaviruses, so they're non-envelope, there's no lipid viral layer, there's only a protein outer strip. And there are also a lot of picornaviruses, or like polio, the the DV68. I'm sorry if I don't remember well, but like other picornaviruses, they're very stable, and so you actually cannot just use like alcohol to disinfect picornaviruses. Or norovirus is actually a good one. You can recover noroviruses from carpets months after after someone vomited on them. So they're very stable, and alcohol doesn't work for them. So you have to use other disinfectants. Vircon, for example, is broad range, a little bit more expensive though. So yeah, just take into consideration that. Okay, so and then also part of biosafety is occupational health. This is an important one because working with animals can be physically, mentally, and emotionally strenuous. And so for occupational health, medical examination and monitoring of the lab personnel will need to make sure that we're not putting people at risk. So going back to safety first, again, research is a privilege. We want to keep people safe when we work. And then for certain respirators, that includes N95s and the PAPR, it does require medical clearance and evaluation before I use them, because it can be strenuous. And also in our lab, for example, we keep our PAPR exposure 4 hours at most, if possible, because it's not, it can be really strenuous to wear that all day. And also consider the potential exposure for zoonotic pathogens, which I think is the next slide. So what are the examples of zoonotic pathogens that you can get for avian species? I think somebody already, like a lot of folks already caught on that. Yeah, salmonella is a big one for avian species. Don't kiss your birds and not wash your hands, and always wash your hands before after you touch if you do have birds or chickens, because they have their poop can have some salmonella. And then Campylobacter is a close one. Also, these two bacteria are found in are normally found in the intestines, the gut of bird species. So yeah, wash your hands. Other zoonotic diseases are chlamydia. So this is I've had a friend who's a pathologist, had to have antibiotic for a couple of months because of exposure to a bird that was infected with chlamydia. No. And then the erysipelathrix is a tongue twister and also a zoonotic pathogen that can infect the skin. You can also get them from birds. Other folks have also mentioned E. coli. That's also another enteric bacteria. And then in terms of viruses, since we are in a VIP lecture, Newcastle and avian influenza are two important ones. Newcastle typically doesn't really cause a very serious disease in humans. It does cause conjunctivitis. That's why there's a picture of an eye with a pink eye there. A lot of poultry are vaccinated with a live vaccine, which means it's a replicating, a weakened form of Newcastle. But even if you get it in the eye, you can get pink eye. So always wear eye protection when you, even if you're just vaccinating birds. Also, I noted here like virulent Newcastle. Another name for Newcastle is avian paramyxovirus 1. Avian paramyxovirus is a select agent by the US Department of Agriculture, and that means that having these agents, you need to register with the select agent program and have a lot more really security, a lot more oversight with how you do research, and also is a reportable disease, and that's because it's a reportable disease to the OIE, because it has potentially big impacts with our poultry industry. And that's the same true with highly pathogenic avian influenza. So there are other strains of Newcastle that are not virulent or low pathogenic avian influenza. Those are not select agents or reportable diseases to the OIE. But I wanted you to know that because this is a virology lecture. Okay, and then in terms of animal biosafety in particular, because animals are unpredictable, their behavior is not always predictable, and they also have dust and dander, which some people are allergic to, so those need to be taken into consideration. So the first time I ever did an animal experiment in higher containment, we were wearing Tyvek suits, we were using isolators, and the first thing that my professor asked me to do is like go open the isolator, and we were wearing PPE, of course. They're the Teletubby suits, so i.e. the Teletubby suits. It's a Tyvek suit with the only holes are for your head and for your hands, for which your head has the or your face, I mean. So like it's just your face and your hands, which are double gloved. Anyway, so the first thing that happens to me when I open the door is a chicken hops on my head and jumps around the room. And so you just never know. And my professor, of course, couldn't stop laughing. And I couldn't stop laughing. Here I'm like super embarrassed. I'm a new PhD student, and I'm trying to chase a chicken in the room that's infected with, it was infected with an exotic infectious bronchitis virus. So yeah, that just takes into, and I'd like to think it wasn't my fault that the chicken jumped out of the isolator, but that's just maybe a funny example of of how we can't always predict animal behavior. So there's a lot of engineering controls that that take into account when you design an animal space. Yes, which I think we'll talk later. Okay, so yeah, I also wanted to mention here, so the Association for Biosafety and Biosecurity has really nice videos on how it is working with animal biosafety level, actually 1, 2, 3, 4. But I only had space for 2 and 3. So I encourage you to go to that website. And that's, yeah. And I know this is the videos that they're showing are mice, and those are mice cages on the left, that the one that are on the racks. But it's pretty much the same principle. So I'm just trying to focus more on the principles. So what are the principles that guide the use of animals? It is kind of different when you are using larger animals like chickens or turkeys. That's a lot different. Which leads me to the next. Is someone raising their hand? OPPOF1S. Are you raising your hands? Maybe not. No. Okay. Maybe just a wrong click. Okay. So which brings me to the next slide, which is the agriculture biosafety level. And this means that, you remember like my story with like the chicken that's loose in the room. So like the actual room is the first barrier. So it's like a biosafety cabinet. So it's the room is engineered so that if the bird escapes, everything is still contained, right? So in this box in a box approach. And here's just some examples of isolators that we use. So like, for example, oh, we're running out of time. Yeah, so so some examples of just isolators, and also the procedure rooms. And you can see in the procedure rooms here, one thing that's super important is the use of directional airflow. So both of these isolators, the inlets, and the inlet meaning the air that's coming in is filtered, and the air that's coming out is filtered, so that the animals, you don't contaminate the animals with potentially pathogens in the air, and the animals that are potentially infected are not also contaminating the environment. That's an important one. And you can also see with the procedure rooms that are the doors are sealed. The doors are sealed, and there's a lot of engineering that comes into play in procedure rooms, especially because it tends to get a little bit messy. Okay, so this is my last slide. So I would like to, these are my take-home messages for you. If you remember nothing else, at least remember the life advice, although it applies to everything else. Begin with the end in mind. So, which means plan your research well in advance, get the knowledge and the skills training needed, and then also seek approval for research activities from your animal care and use committee and your biosafety program. Okay, and then here are my references. Unfortunately, not all of them are open access, but I realized the Avian Disease Manual 7th edition is open access. So, but the 8th one, the most recent one, which is this one, is not. But the rest are open access. So if you want to know more, go ahead, maybe take a screenshot. I'll leave that up for a little while. And I'll entertain any questions if you have any.
Okay, thank you, Dr. Christina, for that very interesting, informative, and interactive lecture. I think our participants enjoyed the activities you provided us.
Okay,
so
great.
With that, we are now open for question and answer. I guess po marami po kayong katanungan, and you may key in your question in our chat box in this Zoom meeting room. And for our audience in YouTube and Facebook, you may also type your questions there in the comment sections. and our moderators will pick them up to be answered by Dr. Layson. So the first question is from Zoom, from Bernard Gutierrez. He is asking if our team would be to do a sampling of a local population of domesticated fowl like chicken, ducks, or etc. And we found a virus of concern. How do we responsibly report the virus without disrupting the livelihoods of those raising the fowl?
Yeah, it's a really good question. Very good question. Yeah, so what do you do? Yeah, so this is the part where you have thinking in advance, and this is great because you're thinking in advance, right? So going back to like you think in advance, what are the possible stuff that needs to be considered? So if you so it depends. Like there is that's always like the I forgot to mention, also wanted to thank Nick Cheplinski, who's our Biosafety officer, and also Anne Hurley Bacon, because I used some of their material, and also Nick gave me some feedback on my slides. Just remembered him. So what he always says is it depends. It always depends. So
Okay.
it always depends on what viruses comes up. So if it's a reportable disease to the OIE, then you have to report it, right? And if it's not, then I think let's just do the not first. So if it's not a reportable disease, you of course have the responsibility to tell the people that that are raising the fowl, and this is a situation, something that is best taken care of in advance. So you already start that conversation before you even start the sampling. So what would happen? How will the data be used, right? So how will the data be used? How will it be shared? Do we need to report it to the state veterinarian? That sort of thing, or to BAI or something. If it's a reportable disease to an OE, then you are required by law to report it. And that comes into play. So, and that's where government programs. So unfortunately, I'm more familiar with the US program, but at least so with the government programs with the US, so let's say you find highly pathogenic avian influenza in your flock, then there are programs, and it's administered by what's called the NPIP, or the National Poultry Improvement Plan. Take note, there's a plan. There's always been a plan. So there's indemnity, what's called indemnity. So to incentivize people to report the disease. So if you report the disease, and number two, follow the procedures that are necessary, so that may involve depopulation, then you will be compensated for every bird that was lost. And that's really important. And that's where government policy really helps, and with the situation, because we need to take care of each other. So yeah, that's my really long question for that. Okay, next question.
Okay. Next question from Brandon Allen Martinez.
Yeah.
For avian research, usually how many samples are required at the minimum?
Okay, yeah, it depends again. I guess it depends. It depends on what the question are. So typically, like I mentioned earlier, with mammalian models, model species like mice, hamsters that are inbred, you can get away with fewer samples that are needed. But if you working with avian species, you typically use more, just because of the type of research that we do. Also, avian species are usually, you do more population based instead of like more individual based type of intervention. So we usually take more, but it depends on what the project is, like what are the minimum samples that are needed. It really depends on the project and the pathogen that are being used.
Okay, thank you. Another question: where can we possibly apply for ethical clearance if an Aya cook is not yet existing in an institution?
Yeah, so that's a good question. I don't know if I have a very good answer for it. However, just because it's like, well, there's no governing body, but maybe also seek the other institutes that have that body, and maybe they can help you out. Because really, like, if we're one, I think it's a good idea. Maybe it's a time to start one. I know which is like a lot of work, but maybe it's time to start one because, for example, it's very it's, I would say, impossible to publish any research from that work if you don't have an IACO in place. So that is a requirement for publishing. And also, it's our responsibility. Also, a lot of bodies that grant that provide research money require you to have one, and so that's something we should have. I'm sorry, that's not a very good answer. That's not a very good answer.
I think that's a very good suggestion, Dr. Leeson. So another question, is there a policy in studying wild birds? Example for O-assist shedding in the environment.
Yeah, wild birds. Yeah, so this includes I know this is that was a very lab
from
centric.
Ryan.
Yeah, from Ryan. Yeah, from PUP. Yes. That was a very lab centric lecture, just because that's what I have most experience with. But we definitely also have samples from wild birds. And then if it's oocyst shedding from the environment, that's usually just fecal material. Like I said, there's really no one policy for everyone, and it's really the institute, the IACOC, and the Biosafety Committee that oversees that. So ask your IACOC and Biosafety Committee whether or not what their policies are. But generally for oocyst, I've worked with oocyst, just wash your hands when you work with them. Wash your hands, change your clothes, take a shower, because you don't want to spread it around. Yeah.
Okay. I think this will be the last question for today. So another question from Eric Roberto Pasqua of the VMUF. In avian research, since you're using them for virus inoculation for potential zoonotic diseases, what are the animal welfare issues do you usually encounter?
Yeah, for animal welfare, it's we'll talk about it actually in the next more detailed, in the more for avian research. I know, like, you can actually apply all of the stuff that I talked about for any research, really. But for zoonotic diseases, the animal welfare issues are usually whether or not what's called the humane endpoints. When do we terminate the animal because it's already suffering? So that sort of thing. And also and the other stuff is we make sure that the animals, because once you have a zoonotic disease, so theoretically the access to that facility is limited, right? So we have to make sure there's no one that's like walking. There's supposed to be no one walking around in that facility. So we have to make sure that we check our birds at least twice a day, make sure that they have food and water and all of that stuff. So the burden of like taking care of the animals will be placed on the researchers, the investigators, because they're the only ones that have access to the facility. That's probably one of the two things that we usually encounter. Yeah.
Okay. So for our participants, huwag po kayong malungkot because at wag po kayong mag-alala, we will be answering your questions tomorrow. So Dr. Leeson will be with us again. On Wednesday, August 18. So we will meet Dr. Leeson again on August 18, 2021, this Wednesday, for the second part of this webinar. So this concludes our first day of our 2-day webinar. Please be reminded that the evaluation form will be given on the next webinar after the second day discussion. So the evaluation form is required for you to secure a copy of the certificate and a copy of the presentations as well. So thank you very much, Dr. Layson, for today. See you on Wednesday po. Same Zoom link, same Facebook page, and same YouTube channel. See you all participants. This has been Madeline Ebarvia of DOST ITDI. Good afternoon again, everyone. Keep safe and God bless.
Okay, thanks, everyone.
Tumitilaok na ang manok, hudyat na ng pagpasok. Paglilingkod na walang kapalit, sa bayan ng aming hatid. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Anumang hamon ay haharapin, mahirap man ay kakayanin. Sa pinagsamang lakas at galing, tagumpay ay mararating. Tara na, kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Ang pangarap ko ay maging piloto.
Pangarap kong magkaroon ng chief alternative for personal hygiene.
Pangarap kong magkaroon ng mabilis at murang transportasyon para sa lahat.
Pangarap kong masagot ang malnutrition.
Pangarap ko pong magkaroon ng effective communication means for emergency.
Pangarap kong ma-maximize yung renewable energy source and to reduce the carbon dioxide emission.
Pangarap ko pong maging scientist.
Ngayon na o simulan na, humanda sabay-sabay aakyat. Hawak kamay tayo'y aangat, lilipad. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Maayos na bukas para sa Pilipinas. bukas ating iparana sa bayang Pilipinas. Ating abutin ang pangarap ni Juan.